So I just want to talk briefly, give you a review of almost 10 years now. We've been working on this area of opening the blood-brain barrier with ultrasound. And just to also show you what our interest is. It is neurodegenerative disease, and I don't think this audience needs any introduction. Just to show you where the coordinates are. So this neurodegenerative disease, especially Alzheimer's and Parkinson's, which are the most common, they hit right in the middle of the brain. So they're very difficult to actually get through just invasive techniques or also even through imaging. But also the fact that, as I'm going to show about focus ultrasound, this is almost a very nice illustration of how having a deep focus, such as ultrasound can do, we can reach these regions. So the main thing, obviously, Alzheimer's hits the entorhinal cortex and hippocampus in its early stages. Parkinson's hits the subthalamic nucleus and substantia nigra, and other less common diseases, but as devastating, such as Huntington's and ALS, also hit the spinal cord and also central areas, subcortical areas of the brain. So why are these diseases treated? There's very smart people, some in this audience, that are trying to figure this out. And one thing that we actually are benefiting is what comes from biology, which is basically a very different neurovascular coupling or vascular coupling happening from the rest of the circulation. So if you were to look at all blood vessels outside the brain in the body, they're very porous. They have a very irregular vessel wall, and pretty much a lot of things can go through the endothelial cell, either between or through them, and reach the surrounding tissue. The actual story is very different in the brain. In fact, everything is very well structured. This is a capillary transverse section. This is the lumen in the middle, and you have the endothelial cell here of the capillary. We have what's called the tight junctions. So there are protein strands that are actually tightening up the endothelial cells. But you also have the pericytes. You also have the surrounding astrocytes. And pretty much what's called the neurovascular coupling is not allowing anything with passive diffusion that's above 400 Daltons. So this is actually a lot of very promising drugs, including some contrast agents, cannot actually penetrate through. So it's a very good system that works almost flawlessly when you have a healthy brain. And if you don't have when you have an unhealthy brain that we actually want to treat. So how do we actually trick that? So there's a lot of other alternatives other than focus ultrasound, but you pretty much have to choose between whether you want an invasive procedure or something that hits the entire brain. For a localized procedure, that would be the direct injection. There's a lot of promise here, a lot of clinical trials, some of them actually in Parkinson's through direct injection. And the idea here is to save the actual rest of the brain from having side effects. Chemically, if you want a non-invasive way of doing it, there's also a way that you can trigger. You can trick some of the transport receptors, or actually change the osmotic effects in the vessels itself, and then you actually do this in the entire brain. So in the region where you want to target, but also in the entire brain. So us and others have actually looked at the focus ultrasound with microbubbles. So I'm going to start with mice, because the mice always work very well for us, and they don't complain. So we basically have put them under anesthesia, and the only thing we do is basically shave the fur. So everything else, scalp and skull, are completely intact. And then we need the water, because ultrasound does travel very well through water, not very well at all in air. So we have this coupling thing, and the concave transducer that you see here, that's around 1.5 megahertz, which is basically something on the order of what's used in cardiac ultrasound imaging. We actually target a small region of the brain. And having all these parameters here, as I'm going to go to the monkey section, we're keeping some of them the same, others not. So it is very easy to go through the mouse skull, but it is a very small brain. So, and we also want to target a specific region in the brain. And for us, we started with Alzheimer's disease, and the area that we targeted was actually the hippocampus. And we like the brain for its symmetry. The contralateral side, or the right side in this case, is actually a control hippocampus on the horizontal section, and the left side is actually what we target. And what you saw here in a very fast movie, I'm not going to run it again for sake of time, is that the gadolinium that usually lines very nicely in an MRI, the ventricles and the vessels, usually does not go to the parenchyma, just because it actually has a molecular weight slightly over the threshold of 400 Daltons, it's around 600 Daltons typically. It can go up to 900. Whereas so the area that we actually targeted, the hippocampus here, actually did let the gadolinium go through, and you can see some of the structures, so somebody can argue that you can use this for imaging, while the contralateral side stays dark. So this actually is another case of a mouse. So this is the histology. This is the actual MRI. We contained most of it around the hippocampus, and then a day later it closed. Same gadolinium injection, the barrier had been reinstated. So how do we do this? So there's the macrobubble is the main thing that we want to introduce here compared to the other two talks. So we have ultrasound, but we also have the macrobubble. The macrobubbles, by the way, have been around for decades in ultrasound. They're used regularly in contrast agents. If you were to get, if you were an overweight patient, you will go for a cardiac ultrasound. Typically, they will not be able to see your heart, mainly because of the fat layers, and they will inject what's called these macrobubble contrast agents, typically. In the US, Definity will be used. And what happens is that they're actually tailored in size to have the specific diameter that will be within the resonance spectrum of the ultrasound frequency used. So they very nicely oscillate, and when we have negative pressures, they actually expand. When we have positive pressures, they tend to contract. So they're oscillating. And this is an illustration of what we think is happening in the blood-brain barrier. So this, again, the magnification and illustration of the capillary. You have the bubbles flowing through. This is the ultrasound going through the actual capillary, and this is the target for us is the neuron. So these bubbles are oscillating, so we can actually do this in vivo. Maybe one day with the brain mapping, we can get to see these bubbles oscillating once in vivo. But for now, we actually have a phantom vessel here that we actually have a BMOS, a standard ultrasound image, pulse echo. Well, we look at two different frequencies, two different pressures. The top one is the lowest pressure where we've seen around which we can see the opening happening. So that's 0.3 megapascal. So that's lower pressures that actually are used in Doppler in diagnostic ultrasound. And 0.9 megapascals where we actually have seen different effects, and I'll show in a minute how this translates to safety. So there is, up here you see enhancement is actually the contrast agents that are doing exactly that. They're enhancing the actual ultrasound response. And you see pretty much over time, as the ultrasound is on here, most of the macrobubbles are actually, there's some radiation force there. You can see them move and oscillate, but not necessarily see them disappear. At 0.9 megapascals, they actually start disappearing. So that's actually something that we have correlated in our papers with what's called inertial cavitation. And the top one is actually called stable cavitation. So instead of the bubble imploding and fragmenting, as you have at point down here, you actually have a stable oscillation that's actually nudging the wall. So that's the idea. And if you were to tailor also the actual size, diameter of the bubble, you're actually able to control the opening. So this is something that we worked with Mark Borden now in University of Colorado, where he was able to sort the bubbles by size. So typically, what's used in the contrast agent, the sizes have a peak around 1 to 2 microns. So the contrast agent ultrasound have a 1 to 2 micron. That again has to do with the frequency that's used in a diagnostic ultrasound. And we were able to contain the opening in one region in the hippocampus. When we actually increase the bubble diameter, everything stays the same. As far as the ultrasound parameters, we're able to actually infuse, in this case, these are fluorescence images. The dextran is actually going through the entire hippocampus. So we're able to actually, through the actual macrobubble, enhance the mechanical effect, stay away from the thermal damage for sure, and then be able to control it also with the area that gets affected. I'm going to go a little bit fast here just to show you that with MRI in 3D, we're able to actually get a bigger region in 3D with large pressure. So the highest pressure we looked in the safety window was 0.6 megapascals and 6 to 8 microns. And we're able to actually see a larger opening area around the hippocampus compared to lower pressures and smaller diameter bubbles. We also use MRI to get the permeability. We wanted to see how the permeability changes. This is something that is done with DC MRI with assuming an arterial input function. And we're able to see that the permeability, so again, in this case, we opened the right hemisphere compared to the left hemisphere. And we could see that the permeability was increasing with pressure in the hippocampus, in the coronal, and also the transverse. And it was hitting a plateau after a while. So between 0.45 and 0.6, we were actually not increasing the permeability. We were increasing the area that actually was permeable around the hippocampus. And we actually compared that with what was happening in the epicranial muscle, which is the muscle that surrounds, that's actually in your scalp. And it was reaching these values here. And at the same time, we were able to also do histological studies. And within that window, we're able to keep this very safe. We didn't see any H&E damage. And just because we always get that question as the previous speaker, so is it safe and what happens to the tissue after you open it? And just to reinforce the safety window that I talked about, if you work around the pressure threshold of 0.3 megapascals, then you compare the two sides of the hippocampi and they're very similar, no damage compared to the control. Then as you ramp up the pressure, at 0.45, we're still very safe. At 0.6, we start seeing red blood cell extravasations 4 hours after sacrifice. And then, of course, if you increase that to 0.75 and 0.9, at the region of the MRI where we had the highest gadolinium enhancement, we were able to actually see dark neurons. So this is basically where, after 4 hours, we just see neuronal damage. Something to say, after we did this study, we also tried to, there were people saying this is short-term damage, what about long-term damage? So we actually sacrificed after 7 days. And some of the damage was actually not there. So this is something that we're still trying to figure out. But it seems that the brain is recovering over time, and this was the acute effect that we found here. This is what I was talking about, the plateau of the permeability over time. So the opening duration is also controlled by the pressure, the pulse length of how long your pulse is, and also the actual macrobubble diameter. So we have found this plateau here with the opening duration for the permeability, but for the volume, the higher the volume that I'm plotting here of the opening that occurred in the brain, the longer it takes to actually reinstate. So it can be on the order of a few hours to several days, as we've seen. Some of the work that we've done also benefits a lot from people in contrast agents in ultrasound. We have actually also looked at nanodroplets that are used from microbubbles, so you can actually condense microbubbles to create nanodroplets, which are on the order of much smaller in diameter, on the order of 200 nanometers now compared to microbubbles. And then once you put them in circulation, because of the ultrasound pressure, they can actually convert to microbubbles, and you can have a more localized effect. And what we've seen at the same pressure, so I'm showing here nanodroplets versus microbubbles, you can get a more localized effect in specific areas of the hippocampus, let's say CA1 and the dentate gyrus, compared to more pronounced effects of the entire hippocampus. And that comes again with the same histology of safety as you have smaller regions of opening. And again, if you reach around 0.6, we start seeing some red blood cell extravasations in the case of the microbubbles. So as far as, again, safety, we used long pulses, or on the order of 5,000 or 10,000 pulses, or the order of 10 milliseconds and 20 milliseconds. So we wanted to see whether that can be done also with short pulses, so closer to what happens in imaging. So you have, ultrasound can go at different frequencies for imaging. The same thing goes for different pressures. So you can go from diagnostic pressure, so very safe, to HIFU and to really high pressures that can cause severe pulverization of the tissue. So we're getting very close to the actual diagnostic now. So in this particular case, we actually targeted again the hippocampus, and we did actually a microsecond type of pulse. I think it was three pulses. And we were able to actually induce opening and have the 3 kilodalton dextran go through. And what we saw actually here was also a more homogeneous distribution. And when we did actually the confocal microscopy of that around the pyramidal neurons, we were able to identify some regions that hopefully make sense to you, to some of the people here, but we thought these were capillaries, and then when we start getting the dimensions, they looked like actually some of the neurons and dendrites that we were getting. So we're still trying to figure this out. This is what we have as a result. So this is a good segue to actual delivery. So of course the main thing is trying to treat the disease. And one of the promising molecules for treating Alzheimer's was BDNF, the brain-derived neurotrophic factor. And we systemically injected mice with that, and we actually fluorescently labeled, in this case with Alexaflor, and we were able to actually see most of the hippocampus that were targeted, especially around the pyramidal neurons that actually uptook the BDNF. There is, of course, endogenous BDNF, and you can probably make out some of the neurons here in the control hippocampus, but this was more substantial. We wanted to see whether this actually was in the neuronal area or was it inside the neuron, so we actually did do some pathway activation staining, and we found this is a MAPK pathway, which is basically determining the protein in the cytoplasm, and where you see brown staining compared to the control area here will be where the actual the BDNF has made it to, the triggering of MAPK pathway has happened in the neurons. And there was another pathway, the AKT, that was also triggered in the hippocampus. And finally, when we put it together, we were able to actually see the actual neuronal pathway be triggered from the actual membrane receptor through 2 pathways, the MAPK and AKT, and finally into the neuronal nucleus. We continue the study with another promising molecule that has hit the clinical trials through AAV delivery called the Nutrain. In this particular in this particular study, we actually targeted the putamen and substantia nigra that are both involved in Parkinson's disease. We were able to, I don't know if you can see that, detected in the putamen that we sonicated with focused ultrasound compared to the contralateral side and the substantia nigra. And when we did similar staining to the BDNF, we were able to identify this inside, in this particular case, in the cytoplasm relative to the control, and inside the actual nucleus. A quantification of that, similar to the BDNF study, at the RET receptor through the 2 pathways in the cytoplasm, and finally quantifying the Nutrain that was found after Elisa inside the actual, in this case, putamen and substantia nigra, was a 200% increase compared to the control side that we found for the Nutrain and relative pathway increase. The last molecule I want to talk to you about is adeno-associated viruses. The AAV1 is the one we actually started with, but I'm going to show you also the AAV2 that after 4 months expresses a GFP, and we're able to detect that in the sonicated side. Again, the brown staining will show you that compared to the contralateral side. And then when we wanted to see, again, which were the actual virus transducted the GFP, this is the GFP signal here with the AAV1 and AAV2 now. And then when we did the neuronal staining and the astrocyte staining, we actually co-localized the expression at the neurons. The AAV2 effect was actually much less, although we could see a few neurons here light up. This is what actually looked at lower magnification. So the control side with the neurons here in red and the astrocytes in blue, we could not see any GFP expression unless the region was actually sonicated, and the GFP here is in green. Okay, the last part I want to share with you is basically trying to make it even harder on ourselves and going to thicker skulls. As Jamie mentioned previously, it does scale with the actual frequency. In our case, we actually use 500 kilohertz. And what we've seen is that the problem does scale very well from mice to primates. And this is very encouraging results. We have very similar parameters otherwise as we actually use in mice. We've used both Definity, which is a commercial agent, and the 45 micron diameter bubbles that we actually make ourselves. We have actually because it's a mechanical effect, we can actually listen to the bubbles similar to the stable cavitation, inertial cavitation that I explained earlier, and we're able to monitor this actually in vivo. So this is an in vivo experiment during opening in a monkey, and we can see the perfusion of the bubbles with stable oscillation as they go in, and you can see that they actually stays. The microbubble perfusion is actually staying pretty stable. These bubbles are very stable. In this particular case, we actually used our own 45 micron bubbles. We can monitor this with the cavitation. The black line that you see is actually the inertial cavitation, something we don't want to happen. At least we want to stay under the regime where the actual bubbles are fragmenting through inertial cavitation. And of course, we check with MRI after the fact for the actual opening. Here, we actually opened a big part of the hippocampus. I'm showing you here some of the cavitation response in the spectrogram. And then the T2 will show whether we have edema or not, so they will actually assess the safety part. A lot of reproducibility on a few monkeys. We have about 6 monkeys now. I'm showing you a few examples of contained opening in the putamen, and also the underlying monitoring results that go with it. The T2s are very clean so far on the monkeys, so no edema that we can actually detect. And as far as the actual targeting, we do have, the skull does actually we know from simulations that it actually does displace the focus. So where we intended it to be in this particular case was actually in the yellow region. The blue region is actually where we ended up having the opening. This is because we don't use any correction in this particular case, and actually in our method. So we are at the mercy of the actual phase aberration, which is very minimal at those pressures to the skull, but there are there. And so through simulations, we actually can predict this axial shift. In this particular case, the total shift was about 6.9 millimeters. The last thing I want to leave you with is that we could target very deep areas and multiple areas. One of them is the calcareous sulcus, the visual cortex, and in the closing timeline, we're still doing these experiments because that requires anesthesia of these primates on a daily basis, which is something that the Animal Care Committee is not happy about. But we have found that within a few days, about 2 to 3 days, the opening has disappeared, in this particular case, in the putamen. So it's a very, very quick summary. We believe that there is a safety window where we can cause just stable oscillation for the opening. The safety window roughly is around 0.45 megapascals for the 1.5 megahertz. This is lower for the monkeys because as you decrease the frequency, the pressure threshold also changes for cavitation. With efficacy, the permeability was increased to a maximum relative to the muscle that lines the actual skull. So we increase the permeability as much as possible as if the barrier was not there, and the barrier did reinstate. And as far as drug delivery, we're very happy with the neurotrophic molecules diffusion, that it actually reached the neurons. And we just still need to do the drug delivery and the primate feasibility that also has been shown to be as safe with the opening, and hopefully get to the clinic at some point. So I want to thank all the great people in my lab and also at Columbia. We have a great team. We have a lot of very good experts in imaging and also microbubbles, and of course our funding. Thank you very much.
I told you it was going to be another great talk. That's really exciting. Thank you very much. Questions?
That was really impressive. What do you think is maybe the biggest challenges from a regulatory standpoint to reach future goal of clinical implementation?
You had to go there.
Just curious.
Yeah, there are big challenges because we have, it's not just a device, it's also a drug, it's also a combo with a drug and also the microbubble. So we were told that this is a nightmare for whoever wants to take this up. But we're not faced. Actually, a couple of days ago, I was talking to our neurologist, Scott Small, we've been collaborating for a long, long time together. And he said, I have these patients coming in and there's nothing to be done. And we can go to the FDA, and there's a lot of people who we hope with the safety that we've shown could potentially benefit without too much risk. So the straight answer is going to be difficult, but hopefully it's not insurmountable. So we'll see. The FDA stuff, so I don't know.
Question? Yes.
Could you clarify, when you oscillate your microbubbles, are they mechanically damaging the vessel tissue, or what is happening when the opening? And what's the size scale of opening you're talking about?
Okay, so it depends on the pressure. So we don't want to mechanically damage the vessels, right? So of course, if you go to higher pressures, you will mechanically damage the vessels, and that's what we see with red blood cell extravasation and also the dark neurons that are supposedly, getting hit by the red blood cells. But there is a window where you have definitely vascular mechanical effects that are intentional. You want to make sure that the vessel responds to the microbubble oscillation, but below the threshold of damage.
So the tension induces opening in between the cells, so the tension induces the opening?
That's the hypothesis. We haven't, so because we can measure the cavitation effects, which are the stable oscillation versus the inertial or collapse of the bubble, we haven't really seen distension of vessels. Again, we don't have the imaging to look at individual capillaries as we wish. We can, especially the subcortical layers. So you could potentially do that on the surface of the cortex, but where we're looking at that resolution, you can't. So it is a hypothesis, putting together that this is what's actually happening, and it's pretty reasonable, we think.
Couldn't you design some in vitro experiment that you wanted to wear?
Yeah. Okay. Yeah. Yes, you can, and there's been people who actually have done that in vitro. Very good work. They have seen dextran being extravasated from the vessel. So in vitro is there. In vitro is confirming what we see. But again, saying that in vitro, what you see in vitro is exactly what's happening in vivo. It's it's not exactly the same. So definitely, the hypothesis has been validated in vitro, but in vivo is it hasn't been validated. So.
The bubbles, they stay in vessels, right? Bubbles will go out.
Yeah, exactly. Yeah, that's a very good question. The bubbles don't. Yeah, that's a very good question. The bubbles don't go through the opening. The opening is actually on the order. It depends on the pressure, and we're looking at that actually. It's on the order of. It's lower than 10 or 20 nanometers. So the bubbles are obviously micron size. Now, if we go to the nano droplets, that also is hundreds of nanometers. So they definitely don't go through. The idea is to become the medium to facilitate the transport. They don't go through themselves.
Next question.
So the drugs are taken up in the first layer of cells, or the day after they penetrate? It's like some operation type of effect. You have a lining of the liver.
Are you talking about the actual bubble being lined with drugs, or not?
Yes. So the drug, the molecule is delivered, right? It's delivered it penetrates the first layer of cells. Is that you consider delivery, or?
Okay, the microbubble and the drug are completely separate here.
Okay, yes.
Right, so the drug is actually much smaller than the microbubble.
Yes.
Right, so the microbubble, so the drug is actually, so that's another good question. The drug is supposed to hit the membrane of the membrane receptors, in this case of the cell, and then trigger that path. It doesn't necessarily have to enter actually the cell. It may not, but it has to go through the blood-brain barrier to hit the membrane of the neuron.
Here?
Yes.
Does that make sense to you? That satisfy you?
So the question is, what happens after that?
After the drug hits the membrane, Absolutely.
I was gonna ask a question related to that. Have you thought about using like a function blocking antibody?
Antibody, yeah.
Function blocking antibody or something that would modulate activity in the brain region, right? I think that would be the question because, I mean, I would assume what happens with MAP kinase pathways would naturally happen in your experiment, right?
I'm not sure exactly what the question is.
How far do they go past the
Okay.
You open the door, but then you want to get them to where you want to get them.
Yeah, so in the case of neutron, that's an excellent question. In the case of neutron, we actually see diffuse in the, in the extracellular space. So we don't know whether it stays there, whether it's cleared out, because, again, this is a snapshot of what's happening. Say, ex vivo, we sacrifice the animal an hour after delivery. So we could do potentially something we haven't done, and we're looking at doing longitudinal studies of sacrificing animals at different time points to see what happens. We haven't done that, but so we haven't done that part.
Yes.
Oh,
Elisa,
yeah.
when you use the nanobubbles, did you pump them at higher frequency?
We actually hit them at exactly the same frequency and exactly the same pressure as we do with the microbubbles.
So, I mean, don't you want you said earlier that you choose the size or the frequency to make sure they oscillate.
Oh, that was for diagnostic ultrasound, actually, the truth. You mean for resonance? Yeah, no, we don't. So I didn't say that correctly. We think the hypothesis there is that the bubbles, the larger bubbles, are more in contact with the, close to the capillary walls because the diameter of the capillary is about 4 to 8 microns. So when you go from 1 to 2 microns to 4 to 5 to 6 to 8, you actually have better contact with closer, the bubbles closer to the capillary wall. So the probability of actually opening the barrier in a larger number of vessels is higher. That's basically our hypothesis. It doesn't tune to the frequency. Because we see that also in monkeys, and the monkeys are 500 kilohertz, we use exactly the same bubble, 500 kilohertz, you have to have much bigger bubbles to actually have the resonance frequency there.
Jamie, you want to ask a question?
So what about like caged compounds or caged delivery? Encapsulating drugs within a bubble and delivering it directly to the tissue. Have you tried that?
Yeah, we had a workshop a couple of weeks ago trying to figure out either coated bubbles or encapsulated drugs in a bubble. We don't have a verdict yet. We still are putting things together. We had very smart chemical engineers and chemists that we were talking to, it's not clear exactly because you will have to, if you have a drug encapsulated bubble, you have to fragment the bubble to release the drug, and you have to do that safely. The drug coating, you also have to fragment the bubble to make sure that that will be. So I think this is another. I think this is something that will be very nice.
You can imagine like the bubble expanding.
Absolutely.
The drug, right?
Yeah, absolutely. Yeah. So we're looking for smart molecules actually, more targeted, and we don't have that yet. So.
Yeah, putting your talk together with the last two talks, could you use, could someone use very, very small bubbles that go through and use it to amplify the response of the neurons to ultrasounds? In other words, like a system.
Yeah.
If you are enhancing the ultrasound effect of the neurons by having a contrast agent.
Yeah. I think I'm not sure if nanodroplets exist in the 10 nanometer or 1 nanometer scale, but if they do, I don't see any difference between that and what we do with the viruses or the dextrans. So that potentially could work.
Any more questions? No, I have one. And you may have mentioned this along the way, forgive me if I missed it. But how about small molecules? The small molecules, like the oxygens and the CO2s. Is the permeability, which of course the vessels are already permeable, does the permeability change significantly? And if so, could this be used as a tool for probing some of the physiology that's a little bit mysterious about that is used in bulb?
Yes.
That's particularly what I'm thinking about.
That's a great idea. That's a great idea. So we believe, this is very simplistic still, that the size that we see, so it's the maximum size. So everything, so for example, let's say we open it at very low pressures, we can open anything that is below 3 kilodaltons goes in. Then that will include also small molecules. And that could have benefits in other areas that so far we haven't looked at. But oxygen has come up also for anesthesia, so that's another.
And there are other anesthetics and things too, but I'm particularly trying, somebody does a little MRI, but not fMRI. I know that exactly what's going on in the bold technique at physiological level is still somewhat mysterious. So if there was another knob to be able to turn to increase or decrease the oxygen flux under certain conditions, in addition to the normal physiological response to increased mental activity, it could be quite interesting.
We're looking at fMRI actually in monkeys.
Collaborate with Truman Brown, so that's...
Yeah. Unfortunately, he left us for South Carolina. We're still collaborating with him, but we're doing fMRI in monkeys because we set up for that, and PET also is something that we're looking at. So we could potentially see that with Truman or others.